Hello, everyone. My name is Stephen Nayfach. I'm a research scientist at the Joint Genome Institute. I'd just like to thank the organizers of Biata for giving me the opportunity to speak to you all today. Today, I'm going to be talking to you about a recent project that we've just finished. It's largely a software project where we've developed a new tool to estimate the quality of metagenome-assembled viral genomes. And the motivation for this project is that there's been really a massive deluge of new viral genomes in recent years. So shown here on the left is a figure from a paper published by Simon Rue in Nature Biotechnology last year, in which he's showing the growth of viral genome databases over time on the x-axis, as a function of the data type. So, and note that the y-axis here is on a log scale. So you can really see that this orange curve, which are these so-called UVIGs, or uncultivated viral genomes, that are derived largely from metagenomes. You can see that these have really increased quite rapidly over time, resulting, when this paper was published, nearly 1 million of these viral genomes from metagenomes. And on the right here, you can now just see a few papers that have been generating a lot of these new viral genomes from metagenomes. Starting in 2016, we had the paper by David Paez-Espino, all the way to more recent papers where massive new genomes from the global ocean, for example. And we fast forward now all the way to today, where in the new release of IMG-VR, which is scheduled for later this year, there's going to be over 2 million viral sequences. So tons of new data. Some of the examples, I guess I would also say that within this massive kind of trove of new sequences, there have been more focused studies that have looked at individual viral clades, including innoviruses, virophages, giant viruses, megaphages, and really the list goes on. This figure here on the right is a figure from a paper by Frederick Schultz, also a research scientist at the JGI, in which he published a paper in Nature earlier this year, in which he mined all available metagenomes at IMG for novel giant viruses. And so this is just a phylogeny of these giant viruses, where you can see the previously known clades are labeled. And so one of the points of this figure was just that this large-scale data mining has vastly expanded the diversity of this individual lineage of viruses. But with these massive, with all this new data, So here is just a slide giving the audience kind of a broad overview of both the experimental and bioinformatics steps to go from a sample to a viral genome. So we start out with our environmental sample from a microbial community. This could be anything from soil to seawater to human stool. And so these environmental samples will typically contain a mixture of viruses and cellular organisms, typically bacteria and archaea, and sometimes microbial eukaryotes. And then the ratio of viruses to cellular organisms can vary quite widely across environment types. In some cases, some experimental protocols will then enrich for viruses, for example, using size filtration, in order to enrich the sample for viruses. But this isn't always done, and this isn't always necessary. So after this step, then we will do the DNA sequencing. Traditionally, this is done using short-read DNA sequencing with, for example, the Illumina sequencing technology, but we could also use some of these newer long-read sequencers. But the focus of today's talk is going to be largely on the short-read technologies. From these short reads, we can then assemble the sequences into longer contigs using tools like Metaspades or MegaHit. But yeah, I should back up for a second and note that even when we do the viral enrichment, as shown here in the second step, this enrichment step is not perfect, and we will still end up with some amount of non-viral DNA, as shown here with these red lines, with these red sequencing reads, and with these red contigs. Okay, so to address that problem, then regardless of whether we've done viral enrichment or not, we can then run computer programs like VirSorter to further enrich our sequences for viruses. And these programs work using a variety of viral signatures, such as viral-specific genes, K-mer frequencies, and things of that nature. And then finally, most studies will then perform some sort of quality control step on these viral sequences. And so the current standard is typically to exclude very short sequences. Sorry, one sec. There's some noise coming from outside. I'm gonna fix that. All right. Sorry about that. The perils of presenting in the time of COVID. Okay, yeah. And so in this final step, we have most studies will do some sort of quality control step, but because there aren't really very good methods to perform quality control at the moment, typically what people will do is simply filter out the short sequences shown here, keeping only contigs longer than, let's say, 5 kb or 10 kb. Or additionally, some studies will further look for sequences that appear to be circular. Okay, and so the focus of this talk again is on these so-called single contig viral genomes. But with these data, there are two main issues to address. So the first one is the issue of completeness. So by taking all sequences, let's say longer than 5 kb, the resulting set of viral contigs can really represent a wide variety of levels of genome completeness. So for example, shown here on the right, let's say our assembled contig is only 5 kb. Well, it might be part of a genome that's actually 50 kb, indicating that this 5 kb contig represents only 10% of the whole genome sequence. And this is really a problem because, I mean, obviously the assembled sequence is going to be missing a lot of the functions present in the full genome, and this can also be a problem when we go to estimate the viral diversity of a data set by clustering the sequences, because many of these very short fragments will fail to align at all, and resulting in an overestimation of the total number of viral OTUs present in a data set. The second issue is shown here on the bottom, and that's the issue of contamination for proviruses. So in many data sets, including those where we haven't filtered out the cellular organisms, there can be the presence of these proviruses, which is just a virus that has integrated itself into a bacterial chromosome. And so these sequences, by their very nature, are not entirely viral. And so after running our viral prediction, we may identify a sequence that is only, maybe has a viral region that's 50 kb, but it's flanked by maybe a short host region that's 5 kb. And this is really a problem when we go to predict the metabolic capacity of the virus, because any of the genes occurring on the host region would be mistakenly assigned to that virus function. Okay, so these are the two main problems that we sought to address. How can we estimate the completeness of these single contig viral genomes? And then how can we estimate the contamination from the host and remove it when present? So we were in part inspired by this tool CheckM, which I'm sure many of the audience, many of the listeners out there will have encountered. And this is really a great tool that has been developed to estimate the completeness of contamination of prokaryotic genomes. And the way this tool works is based on, it estimates the completeness of contamination based on the presence and copy number of these ubiquitous single copy genes. And so a genome is estimated to be complete if it contains a full complement of these ubiquitous genes, and it's not contaminated if all those genes are single copy. And this method has been really widely adopted in our field for prokaryotic genomes, and is extremely useful. But at the current moment, there isn't a corresponding method for viruses, in part because there's a lack of these universal genes in viruses. Okay, and so to address this problem, we developed the tool CheckV, which is a fully automated command line pipeline to estimate both the completeness and contamination of single contig viral genomes. If you want to check it out, here's the link, Bitbucket, so bitbucket.org/berkeleylab/checkv. And so the basic question that this tool asks is, how good is my viral genome? And so what do I mean by good? Well, like I said, in the first step of the pipeline, CheckV will predict whether or not the viral contig contains is flanked by a host region, and will remove that region when present. Secondly, CheckV will estimate how complete is that viral genome. Is it a very short fragment, or is it a near complete viral genome? And then lastly, in some cases, there are signatures of a complete genome sequence. And so CheckV will also look for these signatures and predict complete genomes when present. Okay, so the first thing that I'm going to spend the majority of the time talking about is this estimation of completeness. So how can we, given a viral contig, how can we estimate how complete it is? Well, we developed a very simple pipeline to do this. The pipeline can be largely broken down into two separate stages. And in the first stage, the CheckV runs the so-called AAI, or amino acid identity-based approach, in which it takes the viral contig and predicts proteins on that viral contig. Those proteins are then aligned against a database of proteins from NCBI viral genomes. And based on those alignments, CheckV will then compute the average amino acid identity between the virus and each individual viral genome in the database, and identify the most similar genomes. These genomes, these complete genomes in the reference, will then be used to estimate the expected genome length of our virus, which can then be used to estimate the completeness as a simple ratio of lengths. So in this little toy example here, our viral contig is 70% the length of the top hit in the database. Okay, in some cases, the viral contig may be too novel or maybe too diverged from the reference genomes in order for this AAI-based approach to work well. So to address this, we developed kind of a fallback pipeline called the HMM-based approach. And in this approach, CheckV will uses a database of about 8,700 viral-specific HMMs, and has annotated both the reference genomes with these HMMs as well as your viral contig in question. And what CheckV will do is it will then identify the set of reference genomes in the database that share the same HMM as your viral contig. So in this little toy example here, the viral contig has this, one of the genes is matching this red HMM, and we've then identified these 5 reference genomes that all share that same HMM. Based on this information, then CheckV can then estimate the range of possible values for completeness to get us kind of a so-called 90% confidence interval for the estimated completeness. So while the AAI-based approach gives us a point estimate for the completeness, the HMM-based approach gives us a range. So in the example on the top, the AAI-based approach estimated that our viral contig was 70% complete, but the HMM-based approach estimated that the completeness of the viral contig is between 65 and 80%. So again, the AAI-based approach is very accurate, but only works well when we have a relatively close match. And the HMM-based approach is more sensitive, but less accurate. Okay, so I just mentioned accuracy. So how accurate is this approach? How accurate is this pipeline? So to address this, we constructed a mock data set consisting of 4,000 random fragments from NCBI bacteriophage genomes. Notably, all these genomes were excluded from the database to prevent overly optimistic results. And so after running CheckV and estimating completeness based on the combination of the AAI and HMM-based approaches, we can see that the estimated completeness based on CheckV is extremely well correlated to the true completeness based on the ground truth. So overall, we get an R-squared of 0.96. Using the, based on the estimated completeness values, we can assign these genome fragments to different quality tiers, from complete to high quality to medium quality to low quality. And you can see that again, the true completeness, since this is a simulation, since these are mock fragments, the true completeness is again well correlated with these quality tiers. Okay, so how well now do existing programs perform on this same data set? Excuse me. Well, the only existing program that that estimates the quality of a viral genome, to our knowledge, is this pretty cool new program called Vibrant. So, so Vibrant is a new viral detection method that was just published at Microbiome. And among many other things that the program does is it reports a Vibrant quality score. Again, the complete from complete to high quality to medium quality to low quality. And Vibrant's quality tiers are based on both circularity and the presence of viral marker genes. And comparing these Vibrant quality tiers now to the true completeness, while there was a good correlation, that correlation was overall much worse than that based on either the check V quality tiers or the check V estimated completeness values. So what this shows is that check V's estimated completeness is doing a much better job compared to existing approaches. But I mean, this in some ways this makes sense. Vibrant wasn't specifically developed to estimate viral genome quality, so it makes sense why it wouldn't perform quite as well. Okay. So the next question we wanted to ask was basically how useful is check V for estimating the completeness of viral genomes, particularly novel viral genomes from the environment. Right, so seems to work great for sequences from NCBI, but how well is it actually working when we apply it to real data? So to do this, we applied check V to the IMG VR database. So this database consists of over 700,000 viruses that have been identified from environmentally diverse metagenomes, across all of Earth's biomes. And so first, we estimated the completeness of these viral contigs using the AAI-based approach and the database of reference genomes from NCBI. Okay, and what did we find? So this is a bar plot where the horizontal axis is showing the percentage of contigs from IMG-VR that were similar enough to a reference genome in order to accurately estimate completeness using this AAI-based approach. And so you can see that across different biomes that check V was able to estimate the completeness of between, it looks like 18% and 35% of IMG-VR contigs. Okay, so these results are not super great. This is pretty poor. And so again, the AAI-based approach is based on alignment to these NCBI viral reference genomes. So the question we wanted to ask was, well, how can we expand the diversity of the reference database in order for the tool to be more useful in real data sets? And so to address this, we conducted a global scale search for complete viral genomes from metagenomes. Right, so we wanted to basically expand the diversity of the reference database by identifying complete viral genomes, across all existing metagenomic data sets. Okay, so how did we do this? Well, we used this feature of these direct terminal repeats to identify complete genomes from metagenomes. These direct terminal repeats, or DTRs, are simply a repeated sequence of about 20 base pairs, or 20 base pairs or more, at the start and end of a viral contig. This is a widely used approach for identifying complete viral genomes. It's an indicator of both a circular sequence. These are often just simply referred to as circular genomes, but they can actually also indicate certain replication mechanisms for linear genomes. For example, for headful packaging in Cautiviralis, you will often see these direct terminal repeats as well. And so using this feature, we basically scanned over 14 billion metagenomic contigs from 14 billion assembled contigs from metagenomes and viromes, including all of IMGM, Magnify, and several large-scale published studies of the human microbiome in global ocean virome. So this massive search revealed over 750,000 of these contigs with direct terminal repeats. And without going into too much of the details, we then identified the subset of these contigs that we thought were viruses, we filtered out. We did some filters to remove sequences that we thought could be spurious and not truly complete. And then we clustered these sequences based on 95% ANI to identify a non-redundant subset of 39,000. Okay, so where do these sequences come from? So shown here on the left now is the environmental distribution of these complete genomes. We can see over 14,000 of them came from human gut, but we also get many thousand from other environments such as marine, freshwater, soil, other mammals, plants, other human body sites. I could read all of them, but needless to say, there's quite a bit of environmental diversity amongst these viral genomes. So what taxa do these viruses represent? Well, they largely come from the diverse order of double-stranded DNA viruses from the Cautiviralis. So 27,000 of the 39,000 sequences were annotated to be from this double-stranded, this big group of tail double-stranded DNA phages. But we also identified many other groups, including small single-stranded DNA viruses from the Microviridae, as well as the CRESS DNA viruses, retroviruses, innoviruses, and many other groups. And you can also see the breakdown for each of these taxa, the breakdown of the environments. So for example, from the Microviridae, a lot of these, the vast majority of these came from human gut. Whilst for Cautiviralis, we identified many more from marine environments. Okay, and so finally, as a sanity check, we wanted to compare the genome lengths of these so-called DTR contigs to existing reference genomes from the same taxonomic groups, to essentially make sure that these sequences were not spurious fragments. And so shown here on the right is now just the distribution of genome length for both these DTR contigs, as shown here, as compared to the viral genomes from NCBI. And then they're split up by these different taxa. And so you can see that, broadly speaking, that the genome sizes were quite well correlated and concordant between the DTR sequences and the NCBI sequences, giving us confidence that these contigs are truly complete genomes. Okay, so now going back to the analysis of the IMGVR database. So I showed you this plot before. So again, this figure is showing the percentage of IMGVR contigs that have a completeness estimate using this AAI-based approach when compared to the NCBI reference genomes. Okay, so now the question that we wanted to ask was how do these numbers improve or change when we additionally include in the reference database these new viral genomes that we identified from diverse habitats? That result is shown here. It's pretty cool. We go from about, looks like from about maybe 20% of sequences from IMGVR. We're now increasing that to about, looks like over 70% of sequences from IMGVR. We can get an accurate estimate of their completeness. Just looking at this plot, it looks like terrestrial, so these are soil viruses. Looks like those were maybe the most challenging, whereas sequences from viral sequences from human-associated habitats, we had the best coverage of those. Okay, so these results were pretty encouraging, but still there were about 30% of sequences on average for which we didn't have any estimate of completeness using this AI-based approach against the viral genomes. So what did we do? Well, then we added in the estimates based on the HMM pipeline, which allows us to now basically get a completeness estimate for every single viral contig in IMGVR. Okay, so now the next question is, so now going back, again, this is just showing you an overview of how the tool is working. So on the top, we have the AI-based approach that is using alignments to both NCBI genomes and these DTR viral contigs, and also using this HMM-based approach for sequences that remain highly diverged from the reference. Okay, and so the next question is, okay, so we can estimate the completeness now for IMGVR. How complete are they? Okay, and so on the left now we have a bar plot showing the number of IMGVR contigs that are assigned to different quality tiers. So we have complete genomes, genomes that are over 90% complete, 50 to 90, less than 50, and then sequences that remain undetermined based on both the AAI and HMM-based approaches. And then on the right, you can see the distribution of contig lengths for the contigs in each of these categories. The complete and over 90% complete sequences fall into this broad category of quote-unquote high quality based on the MyUVig standard, and the rest of these fall into the category of so-called genome fragments. Okay, so you can see that on the top, using CheckV, we've identified over 32,000 so-called high-quality genomes from IMGVR. I think this is pretty cool. 32,000. That's a pretty remarkable number. That's more high-quality genomes that are in all of NCBI. However, those high-quality sequences represented a mere 4% of all the sequences in IMGVR. So the majority of the database is represented by these so-called genome fragments that were on average only 21% complete. And the reason for this is it's not specific to anything that IMGVR is doing, but it's really related to the many challenges of assembling viral genomes from short-read metagenomes. And this includes low sequencing coverage for rare viruses, strain heterogeneity. We know that really hampers assembly, as well as things like repetitive regions, etc. And one thing that I just really wanted to plug was that I think one really promising solution to this problem is using long-read sequencing technologies. So shown here on the top are just two really nice studies that have been published, I think, in the past year, which have done exactly that. They have used long-read sequencing technologies to obtain high-quality and complete viral genomes on individual sequencing reads, so without the need for metagenomic assembly whatsoever. And that's because these long sequencing technologies can generate ultra-large sequencing reads that can be even over 100 kb. And again, this really allows us to capture the genomes of viruses from diverse strain populations or viruses that have hard-to-assemble repetitive genomic regions. Okay, so, excuse me. Okay, so going back to this broad, the broad pipeline. So I said at the beginning of the talk that CheckV can be broadly broken down to these three separate stages. Now I'm going to talk a little bit about how CheckV identifies complete genomes. So I already mentioned a little bit that, okay, so to back up a little bit. So like I said at the beginning of the talk, Simon Rue published a really nice paper last year in Nature Biotechnology. And in that paper, they discuss three sequence features that have been used in the literature to identify complete viral genomes. So one of them we've already mentioned. They are these so-called direct terminal repeats, or DTRs, which are often simply referred to as circular sequences. Again, these can indicate a circular genome, but also certain replication mechanisms for linear genomes. And this is definitely the most widely used feature in the field for identifying complete viral genomes. A less commonly used feature are these so-called inverted terminal repeats, where again, the repeat is just simply inverted at the end of the sequence. These are much less common, but have been observed and are believed to be involved in a variety of viral functions, including in replication, recombination, circularization, or genome integration. So they have lots of potential functions, but are less commonly used as a robust signature of a complete viral genome. And then finally, complete genomes can be identified based on either host integration sites or viruses that are flanked on both the 5' and 3' end by host regions. Yeah, and so there are a lot of tools out there that will predict these complete proviruses, including VirSorter, Vibrant, and CheckV will do that as well. Okay, and again, all three of these features have been previously reported in the literature, and I would say are not novel contributions of CheckV. But so we wanted to basically ask the question of how accurate are these three approaches for identifying complete viral genomes from metagenomes? Okay, so how can we address this problem? Well, what we did is we basically used the IMGVR database to address this. So we used these three sequence features to first predict complete genomes from IMGVR. In parallel, we then estimated the completeness of these putative complete genomes based on comparison to closely related reference genomes in the CheckV database using the AAI-based approach, and then compare the results. So what does this look like? So shown here on the left now is the distribution of the estimated completeness, for sequences from IMGVR that had these direct terminal repeats. So you can see that the distribution of completeness is skewed to the right and is very, very close to 100%. So this is reassuring. These DTRs are widely used in the field, and so this is just showing that these DTRs are, we get good concordance between the estimated completeness and the prediction, and so we can say that these DTRs are a pretty robust signature of a complete viral genome. Okay, so what does this results look like for the other types of predictions? So when we look at the ITRs, there were looks like nearly 100-fold fewer ITRs identified in IMGVR, and overall the completeness of those sequences was much lower. So we do see a subset that are close to 100% complete, but also a long tail that range from 0 to about 80% complete. And so these ITRs appear to be a much less reliable indicator of a complete genome. And likewise for prophages, so the prophages were identified based on a viral region that was flanked on both sides by host, as predicted using CheckV. And you can see that these sequences, again, the results were somewhere in between the DTRs and the ITRs. So we see a large number of them that have close to 100%, that are predicted to be 100% complete, but a long tail of lower completeness. But regardless, all three of these approaches were highly enriched for high completeness sequences relative to the background distribution of completeness from all of IMGVR. So shown here is just the distribution of completeness for all other sequences from IMGVR. So I think the conclusion here is that all three of these approaches do have some pretty good signal for complete genomes, but they vary in their usefulness. So one kind of cool biological result is that, so applying this approach to all of IMGVR, we identified a lot of complete viral genomes. The largest complete viral genome we identified was this 528,000 base pair sequence that was derived from a metagenome from an Antarctic lake. And so you can see, looking around, and this massive viral genome had some pretty cool features, including an enrichment of transposases, in a complete CRISPR-Cas system. In fact, this viral genome, it looks like it is a new member of a recently discovered clade of huge phage. And so shown here is an analysis conducted by Simon Root, in which he constructed a phylogeny of these huge phage. And shown here in bold is the placement of this 528 kb sequence relative to these previously published sequences from the Nature paper shown here in the bottom. Okay, so this analysis is basically just to give you a sense of some of the biological results that are attainable using these kinds of approaches. Okay, so finally, in the last few minutes, I'm just going to briefly talk about the last step in the pipeline, where CheckV identifies and removes flanking DNA from the host on integrated proviruses. Okay, so how does CheckV do that? It's pretty simple. Basically, CheckV identifies host virus boundaries using a combination of both gene content, so whether the gene is microbial or viral, as well as GC content. So an example of that is shown here on the right, where we have the list of genes in the viral genome, where the color of the gene indicates the annotation, whether it's annotated to be a microbial gene in green or a viral gene here in blue. And those microbial or viral annotations are based on alignment to a large database of HMMs that we curated from many existing databases shown here on the top. And so based on curation of those databases, we were able to identify a subset of HMMs that were highly specific to microbial genomes, and a subset that were highly specific to viruses. Okay, so again, CheckV identifies these breakpoints where there is basically a big difference in both the GC content and gene annotations between a pair of adjacent windows of genes. Okay, how accurate is this approach? I'm not going to spend too much time on this, but to basically benchmark this approach, we constructed another mock data set of both proviruses in which we took those same bacteriophage genomes from NCBI, which were excluded from the CheckV database, and inserted them into a random location in the predicted host of those phages, the bacterial genomes that were the hosts. And so, so we have these mock data set, these mock proviruses, where we have a viral region flanked by a host region. Okay, and so we ran CheckV to perform this prediction, as well as 4 other tools that were previously published from the literature that also predict these host virus boundaries, including VirSorter, Vibrant, PhySpy, and Figaro. And for all these predictions, we to standardize the predictions across the tools, we said that that a provirus was predicted when a tool said that the predicted viral region covered less than 95% of the sequence length. So using that criteria, CheckV was able to identify about 76, was able to say that 76% of these mock proviruses contained a host region, compared to between 20 to 30% using the other approaches. And so based on this analysis, we concluded that CheckV is significantly more sensitive at identifying these proviruses compared to existing approaches. And that is particularly true for proviruses in which the host region is quite short, or the overall contig length is quite short. So CheckV is particularly good at identifying these sequences that have a short region of host DNA. So how, basically as a negative control, to get a sense of the false positives, we then apply CheckV and existing approaches to a data set of exclusively viral genome fragments. These are sequences that are entirely viral. So overall, CheckV predicted that about 1% of these, that these entirely viral sequences had a host-derived region. So not too bad. VirSorter and Vibrant are actually even more conservative. It had a lower false positive rate. These tools identified only about 0.4% of sequences that were entirely viral. They predicted a host region. So again, these results are all pretty good. In contrast, PhySpy and Figaro, on many of these viral fragments, predicted that the viral region covered only a subset of the contig length. So here we conclude that CheckV is also fairly conservative and doesn't have a high false positive rate. Okay, so finally, how does identification and removal of these host regions affect analysis? Basically, how would this affect your analysis? And so again, we applied CheckV to the IMG-VR database and identified about 40,000 sequences that were flanked on one or both sides by a host region. And so again, these 40,000 sequences represented a very small fraction of the entire data set, indicating that most IMG-VR sequences are entirely viral. But we now wanted to focus on this subset of putative proviruses. So overall, so across different biomes, these host regions accounted for about 12% of the genes on these proviruses. Okay, and so that result is just shown here, where the horizontal axis is indicating the percentage of genes, either from viral regions or host regions, and split up by the biome type. So marine, freshwater, human, host-associated, etc. So basically, regardless of the biome, about 12% of the genes on proviruses are derived from the host. Excuse me. So even on these proviruses, most of the sequence is viral. But we then annotated the genes on those proviruses based on metabolic pathways from the KEGG database. And so now we're not looking at all the genes on these proviruses, we're just looking at the metabolic genes. And what was really striking is that while the host regions only accounted for 12% of the genes overall, they accounted for 42% of the metabolic genes, indicating that if, for example, for analyses looking for auxiliary metabolic genes on viruses, these host regions have a really strong impact on those types of analyses. And finally, looking at individual pathways, individual metabolic pathways within KEGG, we see that the results are even worse for individual pathways like ABC transporters, where over 88% of the genes were derived from the host. And so these results basically just indicate that removing host regions can really make a big impact on an interpretation of viral functions. If you hadn't removed these host regions, you would think that these proviruses had a lot more, for example, ABC transporters than they really had. Okay, and so just to wrap up, today I talked to you a little bit about a new tool, CheckV. It's freely available. The Bitbucket link is on the top. And it basically tries to answer the question of how good is my viral genome in terms of both completeness and contamination. With any tool, there's definitely going to be some limitations, and so I think it's worth just being open about what are the limitations of CheckV. So first, we basically designed CheckV to work for single contig viral genomes. And while most studies are focusing on single contig viral genomes, in some cases it may be necessary to perform binning in order to recover the genome sequence of a virus. For example, for viruses that have extremely large genomes, such as giant viruses, this approach has been shown to be quite useful. But with binning, of course, there's going to be some additional issues, such as the binning process, is not perfect, and so you may have contamination in a bin derived from other viruses and/or other host organisms. And this problem is not currently addressed by CheckV. There may be assembly artifacts that are not detected, such as assembly chimeras. Also, I think it's going to be important to continually update the database in the future to get the broadest coverage of viruses from different biomes and taxonomic groups. In particular, most likely the database is depleted in eukaryotic viruses and RNA viruses. So it's going to be important in the future to update the database as we get a better sense of these types of viruses from different types of samples. And then also segmented viruses pose some additional challenges that are not currently addressed by CheckV. Okay, and so with that, I would just like to say, obviously thank you for your attention. Thank you to my funding sources, and then in particular, thank you to Simon Rue and Antonio Pedro Camargo, who really made some really important contributions, as well as my supervisor, Nikos. And thank you again for your attention.